In 2014, a team of geneticists in Shanghai sequenced the Y chromosomes of nearly 900 Chinese men and found something that should not have been statistically possible. Roughly 40% of them, hundreds of millions of people alive today, could trace their paternal line back to just 3 men who lived between 4,000 and 6,000 years ago. Not 3 tribes, not 3 regions, 3 individual lineages that expanded so successfully they now sit inside the genome of a population larger than North and South America combined. That finding came out of Fudan University, and it was only the opening chapter of a much longer story, one that ancient DNA has spent the last decade quietly rewriting. Scientists wanted to know where the ancestors of modern Chinese populations actually came from. They tested that question against ancient Egypt, against Mesopotamia, against the Indus Valley, against the early farmers of Anatolia and Europe. And when the genetic evidence came back, the ancestry of over 1 billion people traced overwhelmingly to one place, a river basin in what is now northern China, and to farming communities most people outside East Asia have never heard of. To understand what that means, you first need to understand how geneticists read the past out of living blood. DNA does not lie, but it also does not shout. It changes slowly through random mutations that accumulate at roughly predictable rates. When one population splits into 2 and stops interbreeding, their DNA starts drifting apart, mutation by mutation, generation by generation. The longer the separation, the bigger the genetic distance. Geneticists use 2 main types of markers to track this. The first is the Y chromosome, passed from father to son, almost unchanged except for new mutations, which makes it a clean record of paternal lineages. The second is mitochondrial DNA, passed from mother to child, which does the same job for maternal lines. By comparing which versions of these markers, called haplogroups, show up in which populations and how much they've diverged, scientists can reconstruct migration routes, population splits, and expansions that happened thousands of years before anyone wrote anything down. Every non-African population on Earth descends from groups that left Africa somewhere between 50 and 70,000 years ago, based on genetic divergence estimates and the archaeological record of early human sites across Asia and Europe. Those migrants spread east along the Asian coastline and inland across Eurasia, reaching the region that is now China by at least 40,000 years ago. For a long time, this deep human presence in East Asia was firmly established. What remained far? Mercure was the more recent question, the one that actually matters for understanding where modern Chinese populations come from. Which specific ancient communities, out of everyone who has ever lived in or near China, are the direct genetic ancestors of the roughly 1.4 billion people living there today? That question turns out to be harder to answer than it sounds, because ancient DNA is fragile and warm, humid environments like much of southern and central China degrade it quickly. For decades, most of what scientists knew about Chinese population history came from studying living people, comparing haplogroup frequencies across regions and inferring past events from present-day patterns. That approach works, but it's a bit like reconstructing a car crash purely from where the wreckage ended up, without ever seeing the moment of impact. It was only with improvements in ancient DNA extraction and sequencing, techniques originally developed for Neanderthal and Denisovan genomes recovered from cold Siberian caves, that researchers were able to pull usable genetic material out of skeletal remains from Chinese archaeological sites and compare those ancient genomes directly against modern populations, rather than just inferring backward. This is the question that a series of Chinese and international research teams have spent the last 15 years chasing down, using increasingly precise tools. The 2014 Fudan study on 3 founding lineages was one piece, a larger and more consequential piece. He came in 2020, when a team led by Melinda Yang sequenced genome-wide data from 26 ancient individuals recovered from sites across northern and southern China, with remains ranging from roughly 9,500 to just 300 years old. This study, published in the journal Science, did something the earlier Y chromosome work couldn't. It compared whole genomes, not just single lineages, across a 9,000-year time span, and set those ancient genomes directly against DNA from people living in China, Southeast Asia, Siberia, and beyond. Before results like this existed, it was reasonable to wonder whether modern Chinese populations might trace significant ancestry to farming and urban societies elsewhere in Eurasia. This wasn't a fringe question. Through the 20th century, some scholars floated diffusionist ideas suggesting that key technologies and even peoples in East Asia originated in the Near East and spread eastward. Testing that idea properly required exactly the kind of comparison modern ancient DNA methods make possible. Lining up genetic markers from Chinese populations against genetic markers recovered from mummified remains in Egypt, from skeletal material at Sumerian and Akkadian sites in Mesopotamia, from burials associated with the Indus Valley cities of Harappa and Mohenjo-Daro, and from early farming and hunter-gatherer sites across Anatolia and Europe. The results were consistent and, frankly, unsurprising to population geneticists, even if they're revelatory to most everyone else. Ancient Egyptian remains carry haplogroups and genome-wide ancestry profiles that cluster with North African and Near Eastern populations, not East Asian ones. Mesopotamian remains show ancestry tied to the wider Fertile Crescent gene pool. Indus Valley period remains, including genomes published from the site of Rakhigarhi, show ancestry related to early Iranian farmers and South Asian hunter-gatherer groups, with only trace, trade, and contact-level genetic overlap extending toward East Asia. None of these populations show meaningful continuity with the ancestors of modern Han Chinese people. That is not because history conspire had to hide a connection. It's because these were separate human populations that had already been diverging for tens of thousands of years by the time complex societies appeared anywhere, living thousands of miles apart, with only limited, indirect contact through trade networks like the later Silk Road. The real ancestral signal was not found abroad. It was found at home in the Yellow River Basin, among farming communities archaeologists call the Yangshao culture, which flourished from roughly 7,000 to 5,000 years ago across what is now Shaanxi, and Henan provinces. These were not urban civilizations with monument, tile architecture. They built villages of semi-subterranean round and rectangular houses, made distinctive red pottery painted with geometric and animal motifs, and lived on agriculture built around two domesticated crops, foxtail millet and broomcorn millet, which were brought under cultivation, independently of the wheat and barley domesticated in the Fertile Crescent thousands of miles to the west. Later, Yellow River societies, including the Longshan culture that followed Yangshao, added early bronze working, more complex settlement hierarchies, and eventually the oracle bone inscriptions that represent the earliest confirmed form of Chinese writing, dating to the Shang dynasty roughly 3,200 years ago. When researchers compare Y chromosome and genome-wide data from Yangshao era and later Yellow River remains to modern Chinese populations, the continuity is striking. Haplogroup O, and specifically its sublineages O1 and O2, dominate both the ancient Yellow River samples and the Y chromosomes of Han Chinese men today, together accounting for the majority of male lineages across East Asia, a pattern confirmed across multiple independent studies going back over a decade. The 2014 Fudan study traced the three so-called super-grandfather lineages to population expansions that align closely with the Yangshao and Longshan periods, suggesting that a small number of Neolithic farming communities in this region grew explosively, likely as millet agriculture allowed denser, more stable populations to outcompete and absorb surrounding groups. This is a very different mechanism from foreign conquest or migration. It's demographic expansion from within. A follow-up study a few years later, examining Y chromosome haplogroups recovered directly from human remains at Neolithic sites along the Yangtze River, reinforced the same conclusion from a different angle. More than 60% of those ancient samples belong to haplogroup O, closely matching the frequency of that haplogroup in modern East Asian populations generally, evidence that the genetic backbone of the region was already largely in place thousands of years before any of the dynasties historians typically study. And more recent large-scale Y chromosome surveys, drawing on thousands of modern samples alongside newly sequenced ancient genomes, have continued to refine the picture, identifying specific subbranches within haplogroup O that expanded during the Yangshao farming period in the Yellow River Basin, and tracing how those branches later diversified as populations spread outward. One especially striking result comes from a set of late Neolithic remains excavated in the Shaanxi region, close to the traditional heartland of early Chinese legend, where researchers found a genetic profile closely matching modern Han Chinese populations, including a Y chromosome branch that remains one of the most common founding lineages among Han Chinese men today. None of this happened in isolation from what was going on elsewhere in East Asia. A broader synthesis of ancient genomes published in the journal Cell in 2021, led by a large international team, pulled together genome-wide data spanning roughly the last 40,000 years across the region and confirmed that the deepest layer of East Asian ancestry split from other Eurasian lineages tens of thousands of years ago, well before agriculture existed anywhere. What changed around 9,000 to 7,000 years ago wasn't the arrival of a new population from outside East Asia, but the emergence, in at least two separate places, the Yellow River Basin in the north and the Yangtze Basin in the south, of farming communities that grew large and genetically distinct enough to leave a clear signature in the archaeological and genetic record. Everything that followed, including the eventual formation of the population we now call Han Chinese, traces back to the expansion and mixing of those two original farming centers, not to an outside source. But the story doesn't end at the Yellow River, and this is where it gets genuinely interesting rather than just confirmatory. Northern China was never the whole picture. South of the Yangtze River, an entirely separate Neolithic tradition had been developing rice agriculture for even longer, with sites like Hemudu in Zhejiang province showing sophisticated wet rice cultivation, stilt houses, and jade working traditions dating back roughly 7,000 years, with some rice cultivation. Evidence in the wider Yangtze basin pushing considerably further back. Genetically, early Neolithic populations in southern China and northern China were meaningfully distinct from one another, more distinct than northern and southern populations are today, according to the Yang team's 2020 analysis. Early southern Chinese genomes show closer affinity to modern Southeast Asian and Austronesian-speaking populations than to northern Chinese ones, supporting the idea that southern China, and specifically the region around the Yangtze and the Taiwan Strait, was the ancestral homeland from which the ancestors of today's Austronesian-speaking peoples, who now stretch from Madagascar to Easter Island, eventually dispersed. Makeup of southern China, reducing the genetic differentiation between north and south that had existed thousands of years earlier. This wasn't a single invasion or clean replacement. It played out over an extended period, likely tracking the southward spread of rice and millet agriculture, state formation, and, later, the deliberate settlement policies of successive Chinese dynasties. The genetic result today is a gradient. Northern Chinese populations carry the highest proportion of Yellow River-associated ancestry, while southern populations carry a blend of that northern ancestry mixed with the older, distinct southern lineage, with the exact proportions varying by province and correlating closely with geography. This southward expansion also helps explain one of the more surprising findings to come out of ancient DNA work in the region, the origin of the Austronesian language family, which today spans a wider geographic range than almost any other language family on Earth, from Madagascar off the coast of East Africa to Easter Island in the Eastern Pacific, and includes languages spoken across Taiwan, the Philippines, Indonesia, and much of the Pacific Islands. Linguists had long suspected, based on vocabulary and grammar reconstructions, that this entire language family originated in or near Taiwan. The ancient genomes recovered from Neolithic sites along the southeastern Chinese coast, and the Taiwan Strait Islands back this up directly, showing a clear genetic connection between early farming populations in that region and both ancient and modern Austronesian-speaking peoples. In other words, the same genetic transition that reshaped southern China internally, the spread of northern-associated farming ancestry mixing with older southern lineages, also set the stage for one of the largest and most geographically dispersed language and population expansions in human history, one that unfolded almost entirely by sea rather than by land. The picture in the far south and southwest adds another layer. Populations in provinces like Guangdong, Guangxi, and Yunnan today carry a mix of northern Yellow River ancestry, older southern lineages related to the deep Yangtze and coastal populations, and, in the southwest specifically, additional genetic input from highland and Southeast Asian adjacent groups that had been living in the mountainous border regions for a very long time. Researchers studying present-day minority populations in these areas, including groups like the Qiang in Sichuan, have found genetic signatures that trace back to early Yellow River-associated ancestry, layered with later, more localized admixture, painting a picture of gradual repeated contact and mixing across thousands of years, rather than a single wave of settlement. A separate and equally telling case comes from the Tibetan Plateau, and here the science moves from population genetics into something even stranger, human interbreeding with an extinct hominin species. Tibetan populations carry a distinctive version of a gene called EPAS1 that helps regulate the body's response to low oxygen, allowing Tibetans to live at altitudes above 4,000 meters without the dangerously elevated red blood cell counts that affect most lowland humans who move there. In 2014, a study published in Nature found that this beneficial version of EPAS1 has an unusual structure that doesn't match anything else in the human genome, except for one thing, the genome of the Denisovans, an extinct human relative known mainly from a handful of bone fragments found in a Siberian cave. The conclusion, confirmed and refined by subsequent studies, is that ancestors of modern Tibetans interbred with a Denisova Put together, these studies describe something real and worth taking seriously, even without exaggeration. An unusually high degree of genetic continuity in East Asia, stretching from Neolithic farming communities through to the present, that has persisted across roughly 7,000 years of dynastic change, foreign conquest, and demographic upheaval. This becomes clearer when you compare it to Europe over the same span. European population history has been marked by well-documented large-scale genetic turnover. Around 5,000 years ago, groups of Yamnaya steppe pastoralists, originating in the region north of the Black and Caspian Seas, migrated into Europe in large numbers. Ancient DNA studies led by researchers, including David Reich's lab at Harvard, have shown that this migration replaced a very large share of the existing Y chromosome diversity in parts of northern and central Europe within a few centuries, alongside the earlier arrival of Anatolian farming populations that had already reshaped the genetic landscape a couple of thousand years before that. Britain saw a further striking turnover around 4,500 years ago with the arrival of Bell-Beaker-associated populations, which genetic studies estimate replaced somewhere in the range of 90% of the existing gene pool on the island within a relatively short window. Modern Europeans, genetically speaking, are the product of at least three major, partially replacing migration waves layered on top of each other: original hunter-gatherers, early farmers from Anatolia, and steppe pastoralists from the Pontic-Caspian region. China's genetic record shows nothing on that scale. Even the Mongol conquest of the 13th century, which placed China under foreign rule for nearly a century under the Yuan dynasty, left a comparatively modest genetic footprint. Studies estimate that conquest that established the Qing dynasty in the 17th century, and for the earlier centuries of contact and occasional conflict with steppe confederations like the Xiongnu, the Xianbei, and later the Khitan and Jurchen states along China's northern frontier. Each of these groups left some genetic trace, particularly in northern Chinese populations closest to the frontier zones, but none of them approached the scale of demographic replacement documented in Bronze Age Britain or Central Europe. Political control changed hands more than once across Chinese history, but the underlying population and the agricultural base, writing system, and cultural institutions built on top of it proved remarkably durable. Ancient DNA recovered from burial sites spanning multiple dynasties reflects this continuity directly, rather than just implying it. Genomes from tombs dating to the Han Dynasty, roughly 2,000 years ago, align closely with the genetic profile of modern northern Chinese populations. Genomes recovered from sites associated with the Shang Dynasty, more than 3,000 years ago, show the same pattern, including in at least one well-documented case, a Y chromosome lineage that remains common among Han Chinese men today. Put simply, if you compared the genome of an ordinary farmer living along the Yellow River during the Shang dynasty to the genome of someone living in the same region today, the differences would be modest, especially set against the scale of genetic turnover that separates ancient and modern populations in much of Europe over the same stretch of time. None of this is unique to China in principle. Population geneticists see similar, if less extreme, patterns of continuity in other regions with comparable geography and agricultural depth, including parts of the Fertile Crescent itself, where farming populations have left a genetic imprint on the modern Middle East that long predates any of the empires that later rose and fell there. What makes the Chinese case stand out is the combination of factors involved and how consistently the genetic evidence lines up with the archaeological and historical record across such a long span of time. Several converging factors likely explain this. Geography is the most obvious one. The Yellow River Basin and the North China Plain sit behind a formidable set of natural barriers. The Himalayas and the Tibetan Plateau to the southwest, the Gobi and Taklamakan deserts to the north and northwest, and mountainous, difficult terrain across much of the southwest. These barriers didn't make China immune to invasion, but they did limit the frequency and scale of the kind of large, sustained population movements that reshaped ancient Europe. Agriculture mattered too. Millet farming in the north and rice farming in the south supported dense, settled populations from a very early date, and dense Ural populations are demographically harder to displace or outbreed than the sparser pastoralist groups occupying the steppe to the north. And cultural and institutional continuity reinforced the genetic pattern rather than the other way around. A shared writing system that remained legible across regional spoken dialects, and a civil service examination system that rewarded male. Study of a shared body of texts created strong incentives for non-Han elites, including Mongol and Manchu ruling classes, to assimilate culturally and intermarry into the existing population rather than remain separate. Over a few generations, distinct conquest-era lineages tended to be genetically absorbed rather than replacing what came before. None of this means Chinese civilization was untouched by outside contact, or that its population has been static. The genetic and archaeological record shows plenty of movement, mixture, and exchange, from the northward and southward flow of ancestry within East Asia itself to genuine contact, along overland and maritime trade routes that connected China to South Asia, Central Asia, and eventually further west. What the evidence does not support is the idea that Chinese civilization, its writing system, its agriculture, or its foundational population originated somewhere else and was imported wholesale. Chinese writing developed from an independent tradition with no structural relationship to cuneiform or hieroglyphics. Chinese agriculture centered on crops domesticated locally, not borrowed from the Fertile Crescent package of wheat and barley, and the population carrying that culture forward traces, according to every genetic study conducted on the question so far, overwhelmingly to farming communities that lived along the Yellow and Yangtze rivers, not to Egypt, not to Mesopotamia, and not to the Indus Valley. It's worth being honest about what this discovery is and isn't. It is not in itself a shocking twist that upends established history, because no serious historian or geneticist ever expected ancient Egyptians or Mesopotamians to be the direct ancestors of modern East Asians in the first place. Populations that formed and diverged over tens of thousands of years in different parts of the world simply don't work that way. What the research actually demonstrates, and what makes it genuinely significant, is something more specific: the unusual depth and durability of genetic continuity in East Asia compared to other major world regions, the surprisingly early and independent domestication of millet and rice as separate agricultural inventions from the wheat and barley package developed in the Middle East, and hard genetic confirmation of something archaeologists had already suspected from the material record, that Chinese civilization is one of a small number of places on Earth where agriculture, writing, and complex society all developed locally without being diffused in from somewhere else. Mesopotamia, Egypt, the Indus Valley, Mesoamerica, and the Yellow and Yangtze River basins now stand as separate, independently verified cradles of civilization, each built by populations that developed complex society on their own terms, using their own crops, their own technologies, and their own writing systems, without needing to borrow the idea from anyone else. That reframing matters more than any single sensational claim could. For much of the 20th century, a diffusionist model of history treated a handful of Old World centers, Mesopotamia chief among them, as the source from which civilization spread outward, with everywhere else assumed to be a later derivative copy. Genetic and archaeological evidence has been steadily dismantling that assumption for decades, not just for China, but for Mesoamerica and other independent centers of early complex society as well. The Yellow and Yangtze River evidence is one of the clearest and best documented cases. A population that expanded from within, developed agriculture, writing, and statecraft independently, absorbed rather than replaced its neighbors over thousands of years, and remains genetically traceable in overwhelming proportion in the people living in that same region today. Given everything ancient DNA research has revealed just in the last decade about the Denisovan legacy hidden inside Tibetan physiology, about the deep north, south divide that shaped early China, about the steppe migrations that erased so much of ancient Europe's genetic history, it's clear this field is still uncovering stories nobody had access to even 15 years ago. If this kind of deep history science interests you, subscribe. Because there are a lot more of these stories to tell, and turning genetic data into a clear, accurate picture of the human past is exactly the kind of work this channel exists to cover.